Number one, if I, oh, you can have my microphone.
Ouch.
Okay, so I have a first question for Dr. Kobal. Have you looked into the excess GAA in the lysosomal health? Because the virus is going to produce like continuously. In the cell, usually there is on and off switch. But if you introduce virus in the body, it will produce continuously GAA. And what will happen with the excess GAA in the cellular system?
Yeah. So it's interesting. I mean, another way to look at that, a question would be, is there toxicity from overproducing GAA? And there's a lot of data from ERT that says that huge amounts of GAA protein injected on a regular basis are non-toxic. And so our vectors actually, in the current doses, reaching more physiologic levels in muscle or slightly above, and there's no toxicity. And we've done formal pharmacology toxicology studies as required by FDA and seen no toxicity. So it's a hypothetical question. Would there be a problem from having too much GA protein around more than normal? But there doesn't seem to be any damage from it so far.
Yeah, it's transgenic, overexpressing GAA in skeletal muscle on the normal background that high.
That too.
Okay, we can worry about too much, but for the time being, we have too little. So, any other question?
One more question for Dr. Byrne. How long patient will need immunomodulators? Like you are talking about
Yeah, it's only one induction therapy right before the dose of the vector's given. So it's really only lasts about 3 months.
And then that's it? They don't need a whole life? Okay.
If there are any questions from the patients, I'll give them priority. But if they're over there,
Yeah.
Is there a microphone, Thomas? Sorry, but
Thank you. It's just a quick question for Frank. I wanted to know who's funding your study. Is it Amicus or Genzyme?
Frank Martineau? Who's Frank?
Sorry, I know it's not.
Is
I know
any
Frank's
one
not
of
so
you
bad.
sponsored by Amicus or Genzyme or yes or no? No, no. Answer is no. No one is sponsored.
No, Frank. I meant. Sorry.
Okay, now.
I know. Sorry, I do apologise. I'm still behind. I'm catching up.
Yeah, my question is for Dr. Koval. Can you say how much GAA production increased in your cohort 1 participants after gene therapy?
I'm not prepared to show that data today. And I think you have to realize, as mentioned earlier, we're in partnership with an industry sponsor, and so we have to be very careful about sharing the data, but we will in due time.
Can you say at what point you think a person would hit their max on GAA production?
Oh, I mean, very generally, we understand the timing for the production of protein from an AAV vector, and it's within a couple of months of giving the vector. Yeah.
Thank you.
Over there.
I know that the trial of Sparks is beginning, and I know that the inclusion criteria is about 6-minute work test and functional respiration. Why, if people can't do the 6-minute work test, can't have the possibility to have the trial?
I think the question is for me, but in fact it's not for me. Yeah, probably you need to address this question more to a doctor, because one, I'm not sponsored from SPAC.
Right. So we're using this kind of criterion in the initial phase 1 study and probably a subsequent one, and we do need the 6-minute walk test data for the FDA, and so in the initial trials, I'm afraid that those kind of criteria will be part of most studies.
But your question's a good one, and it's reasonable to imagine that there's an independent study. So when you mix the two things together into one study, it becomes confusing. But if there were an independent study that focused merely on respiratory function, then ambulatory status wouldn't actually matter conceivably. And this is being considered also in the Duchenne population to work to include non-ambulant patients as well.
Would a study like that with non-ambulatory patients possibly
Certainly
transfer
every
over to
study is
infantile?
tailored to have outcome measures that match the patient population that are being studied. So an infantile onset study would be specifically structured around both those safety and efficacy outcome measures that would be relevant to infants.
But if you, then lady first.
So I have a question for you, Barry. I think this is very interesting work, but I was curious because your injections are intramuscular, and we know that the immune response is not as robust when you do an intramuscular versus an intravenous for the re-administration. So is that something that has also been done?
Yeah, there are the same animal groups, both systemic and intramuscular, but the redosing experiment in the primates was only IM. So, but in fact, like the reason we give immunizations intramuscularly, the response is actually greater from the intramuscular administration because the vector all reaches the draining lymph nodes. So, but nonetheless, the same findings when it's given intravenously.
And I had another question. I saw a slide up there with the combination of 3-drug versus 2-drug regimen. And so have you seen any differences? Or
No, it's interesting. At least in rodents, the Velcade was not as beneficial as we would have expected from the human experience, particularly the work you've done with ERT. And it's not well tolerated by rodents. So it remains to be seen whether just the CD20 rapamycin approach will be sufficient, but there are additional antibodies that actually are non-depleting that we're also investigating that will probably have a similar effect that may be useful. I think one of the key takeaways that was surprising to us when we actually have now, in addition to the human study with the in Pompe with the IM dosing, we have 2 other indications where the strategy's been used now in 3 patients. And what is most striking to us, since you can't do these experiments very well in primates, if you do a careful PK of the viral distribution in the blood, the absence of antibodies actually prolongs the exposure in the blood and increases the efficacy about 4-fold. So we expect the relative dose to be higher. I mean, much like clearance of any anti-drug antibody affects the drug clearance. I saw another question coming from somewhere.
Yes, sorry,
Pascal.
it's not for, I have a question for Dwight and Giuseppe regarding the gene therapy trials. If I am not wrong, in both trials, Sparks and Duke, the patient remain treated with myozyme when receiving the gene therapy. And my concern is how will you assess the benefit, the clinical benefit of gene therapy, knowing that the patient still receives the enzyme? When will you decide to stop enzyme therapy? So my question is about the clinical efficacy.
I'll answer about our trial, and yeah, we do have criteria for withdrawing ERT, and so based on the patient's response in the clinical trial, we'll withdraw ERT, and then we can see the benefit of the gene therapy as a standalone.
I had a question, and Barry indicated that about 50 to 60% of the patients has AAV gene antibodies against AAV. Is that also your experience? Did you have to screen a lot of patients to find patients for this trial? And I also wanted to ask, did you also think that ERT antibodies are relevant for the efficacy of the therapy?
Right. Yeah, those are 2 really good questions, and it seems like the prevalence of anti-AAV8 antibodies is similar for the Pompe patient population as seen in other populations, about 40 or 50% for adults. About the anti-GA antibodies, this immune tolerance induction actually should reduce those anti-GA antibodies. So there's a potential for a patient who already has high titer antibodies to be tolerized to GAA by getting this gene therapy.
So are patients with anti-GA antibodies excluded from the study currently?
So we have a cutoff, a conservative cutoff in the phase 1 to prevent high titer antibodies interfering, but we're following it as an endpoint, and that probably later we wouldn't worry about the anti-GA.
No, but that doesn't come through this way. Because I can't hear it, so no one else can hear it either, I think.
I wanted to respond to the same question, because I think there are 3
Oh, okay, so we want an answer from
Yeah, Giuseppe as
Dr.
well.
Marchetti
Yeah, on the
sorry,
same
sorry.
answer, on the same question.
Yeah, the first one, the one from Pascal, was about the eventual suspension of the ERT. I have no experience on this specific disease. I have experience with Crigler-Najjar. We are doing a clinical trial on Crigler-Najjar. In that case, the treatment is light irradiation. And in fact, the decision was made that would be depending on the results that you have during the trial. So maybe that's why we are developing all this kind of comparison with ERT and so on. Depending on the results that you have during this trial, you decide the criteria that you want to apply to before you remove the ERT. In that case, was the light, and so I imagine that the relevance of the data I showed you is because they can give you a criteria to stop the ERT, knowing that in the case of Crigler-Najjar, stopping ERT means that the patients can die. In the case of Pompe disease and LOPD, stopping ERT is not a big issue.
It probably becomes challenging to do this in the context of an open label study, though. At some point, these interventions need to be blinded, both to the treatment and to the concomitant treatment.
One last question from the back.
Yes, this is for Dr. Byrne, and I hope this is for the benefit for everybody. I'm still very blurred about the phrenic nerve and pomp and respiratory function. Could you give 1 minute, if possible, if we have enough time, explanation of the link between the phrenic and the rest of the respiratory?
Yeah, there are a couple of lines of evidence related to all lower motor neurons, so both the cranial nerves and in the spinal lower motor neurons, that there are signals that suggest that over time those neurons are lost through cell death, that one can compensate for that up to a point, and then that's probably the point at which independent ventilation is lost, because there's a lot of redundancy in the system, but not enough to withstand a complete loss of all the motor neurons. So that'll be one of the key indicators for early intervention, because we have enough redundancy in that circuit to prevent loss of respiration with aging. There's some lower rate of neuronal loss as well, but this is something we think is an important rationale for the early treatment.
Thank you.